Today's topic for discussion is industrial applications of microorganisms. Let us see all the details of industrial applications of microorganisms. The industrial microbiology is concerned with all the aspects of business that is related to microbes. The industrial microbiology is concerned with number 1. Employing microorganisms to produce desired products, preventing microbes from diminishing the economic value of various products. Several organic acids are produced, amino acids are produced from microorganisms, enzymes, alcohols, organic solvents, synthetic fuel is produced from microorganisms, and recovery of metals from ores through bioleaching is also done by the use of microorganism. Recovery of petrol and single cell protein production is also done by the use of microorganisms. Industrial microbiology is an important area of applied microbiology. It refers to the use of microorganisms in commercial enterprises. Cheap raw material are converted to valuable products through the metabolism of microbes. Microbes for this purpose could be exploited in different ways. For instance, this include synthesis of fermentation products as acid in another desired types, the production of enzymes, antibiotics or insecticides, the use of microbes themselves as food. The alcoholic beverages are prepared by fermentation process. The fermentation is industrial microbiology, is used in a wider sense to include any chemical transformation of organic compounds carried out by using microbes and their enzymes. The process is non-specific. Next, the microorganisms helps in cheese making, lactic acid in curd, CO2 in bread and alcohol in beer and wines. Yeast is cultivated in large scale for brewing and baking industries. Efforts were made to develop means of stabilizing large quantities of media, fermentation vessels and the pipelines. Steam was often used for this purpose. The growth media were often byproducts of the molasses and yeast extract. Most of the products come from anaerobic microbial fermentation, that is acetone and alcohol, and hence the industry that grew up was known as fermentation industry. Next, in industry, the large quantity of culture of microbes is done under aerobic conditions and aseptic state. The strains of the microbes that are more efficient in antibiotic production were selected. Strains of penicillium in current use produce as much as 10,000 times as much penicillium per unit volume of fermentation liquid than does Fleming's original isolate. During the 1960s, the microorganisms are used as bulk sources of protein for animal and human food. The so-called single-cell protein led to exploitation of continuous culture method on industrial scale. This requires the development of sophisticated fermentation equipment and the complete aseptic condition. The most recent development in the biotechnology, the term often being used indiscriminately and fashionably. It can be defined as the application of biological organism systems or processes to the manufacturing and service industries. These have been some tendency to equate gene cloning and recombinant DNA technology with biotechnology, though other many areas are included in it. Other important areas are the use of enzymes and immobilized cells as catalysts and sensors, the manipulation of cell physiology for the maximum production of metabolites, and the use of cell fusion to generate either monoclonal antibodies or new cell lines, etc. Microbes as ideal organisms. Microorganisms possess several characteristics which make them most ideal organisms for industrial purpose. They possess a broad variety of enzymes making an array of chemical conservations possible. They have a relatively high metabolic activity which allow conservations to take place rapidly. They possess large surface areas for quick absorption of nutrients and release the end products. Moreover, they have a high rate of multiplication as a 20-minute generation time for E. coli under ideal condition. In an industrial process, a microorganism in fact represents a mini chemical factory. The efficiency of this factory depends upon the liberation of large amount of a single product that may be efficiently isolated and purified. The microbe should be easily cultivated and maintained and should have genetic stability with infrequent mutations. Its value enhances if it can grow on inexpensive, rapidly available substrate that may be the byproduct of other industrial processes. For example, a large amount of whey is produced in cheese production. If there are other microbes which could convert the whey components to lactic acid, this will add to an overall profit of cheese industry. Next. There are many species of microbes which are used for production of commercial products. Some species which are used for production of commercial products are shown below. That is, ethanol is prepared from glucose by the use of Saccharomyces cerevisiae, and acetone and butanol is also prepared from by the use of Aspergillus niger, etc. Amino acids are prepared, amino acids, lysine and linoleic acid, these are the amino acids. Amino acids are prepared from Corynebacterium, etc. Vitamins like riboflavin, vitamin B12, these are prepared from several types of microorganisms. Next, enzymes are prepared from amylase, it is the enzyme, glucoamylase, cellulase, invertase, these are the enzymes which are produced from different microorganisms like Aspergillus oryzae, Aspergillus niger, Trichoderma, Saccharomyces, etc. Polysaccharides like dextran and xanthine gum, it is produced by the microorganisms again like Xanthomonas, Leuconostoc, etc. Next. Pharmaceuticals, in this case penicillin is produced from Penicillium species, cephalosporin produced from Cephalosporium, and gramicidin produced from Bacillus species, Bactracti, it is produced again Bacillus from Bacillus, Mycobacterium, it produces steroids, helps in steroid transformation. Arthrobacter, that also helps in steroid transformation. E. coli, that produces insulin, human growth hormone, etc. Next. Antibiotics are also produced for microorganisms. Pharmaceutical manufacturing industry is a major source of employment for industrial microbiologists. Microbes have maximum applications in medicines, which include antibiotics, steroids, vitamins, and vaccines. Next. These are the different antibiotics which are produced from different types of microorganisms. Next. Penicillin. Inoculums of penicillin Chrysogenum is produced by inoculating dense pore suspension into a wheat bran nutrient solution, and that is incubated at 24 degrees Celsius temperature for 1 week or so on, and then transferred to a inoculum tank. Tanks are agitated with forced aeration for 1 to 2 days to have heavy mycelial growth for subsequent inoculation into a production tank. The typical medium used for propagating penicillium today has total glucose and molasses. The pH of the medium after sterilization must be 6.0, and this procedure that takes 7 days. for fermentation, complete fermentation. After fermentation, if the fermentation is completed, the concentration of penicillin reached at the maximum achievable level, the liquid medium containing penicillin is separated from the fungal cell using a rotating vacuum filter. Fungus biomass is scraped from the surface of the filter drum, dried and marked as the animal feed supplement. Penicillin is discovered from the filtrate using various extraction. Penicillin is recovered from the filtrate using various extraction methods. Cephalosporin. Cephalosporin is made as the fermentation product of Cephalosporium aramonium. However, this form is not potent for clinical use. Its molecule can be transformed by removal of an alpha amino adipic acid side chain to form can be further modified by adding side chain to clinically, it is useful broad spectrum antimicrobial. Various side chains can be added to as well as removed from both 6-APA and 7-ACA to produce antibodies with varying spectra of activities and varying degree of resistance to inactivation by enzymes produced by the pathogenic organism. Thus, we have entered into the so-called third generation cephalosporin such as moxalactam developed for control of bacteria that produce enzymes capable of degrading penicillin and cephalosporins. Next, streptomycin. Streptomycin and various other antibiotics are produced by using strain of Streptomycin griseus. Spores of this actinomycete are introduced into a medium to establish culture with a high mycelial biomass for introduction into an inoculum tank with subsequent use of the mycelial inoculum to initiate the fermentation process in the production tank. A classic fermentation process involves three phases. During first phase, there is a rapid growth of microbes and that is rapid growth of microbe takes place and proteolytic activity of the microbe release NH3 to the medium from the soybean meal causing a rise in pH. During this initial fermentation phase, there is a little additional production of mycelium, but the secondary metabolite streptomycin accumulates in the medium. The glucose and NH3 releases are consumed during this phase. The pH remains fairly constant between 7.6 and 8.0. In the third and final phase, when the carbohydrate become depleted, streptomycin production cases and microbial cells being to lyse. The pH increases and process normally ends by this time. After the process is complete, mycelium is separated from both by filtration and the antibiotics recovered. In one method of the recovery and purification, streptomycin is absorbed into the activated charcoal and eluted with acid alcohol. It is then precipitated with acetone and further purified by use of column chromatography. Next, steroids. Microbial biotransformation of steroid is very important in the pharmaceutical industry. Steroids are used in treatment of various disorders and also involved in regulation of sexuality. The fungus Rhizopus nigricans, for example, hydrolyzes progesterone, forming another steroid with the introduction of oxygen at the number 11 position. The use of such microbial biotransformation in the formation of cortisone has lowered the original cost of 4,000-fold. Human protein genetic engineering has expanded the industrial applications of microorganisms, including production of human proteins. By using recombinant DNA technology, human DNA sequences that code for various proteins have been incorporated into the genomes of bacteria. Vaccines. Production of vaccines. Vaccines are also produced by the use of microorganisms. Production of vaccines involves growing the microorganism, processing the antibiotic, antigenetic properties needed to elicit a primary immune response. Mutant strains or inactivated virulent pathogens are used for producing vaccines. Vitamins. Vitamins are also produced by the use of microorganisms. Microbial fermentations are used for commercial production of several essential animal vitamins. Some important vitamins and their microbial sources are shown below. That is, riboflavin is prepared from Asba gossypi. Sarbose is prepared from Gluconobacter oxidans. Vitamin B12 is prepared from Propionibacterium shermani. Okay, next. Organic acids are also prepared from by the use of microorganisms. Several organic acids including acetic acid, lactic acid, fumaric acid, gluconic acid are prepared from by the use of microorganisms. Gluconic acid is also prepared from by the use of microorganism. That is, in this case, bacterium is used. That is, acetobacterium is used for the production of gluconic acid, and several fungi, aspergillus and penicillium, these are also the sources of gluconic acid. Citric acid is produced by the use of microorganism. It is produced by Aspergillus niger. Citric acid is used as food additive, especially in soft drinks as a metal chelating and this is commonly used, and it is essential to limit the growth of the fungus so that high levels of citric acid can accumulate magnesium sulfate and potassium sulfate. Next. Itaconic acid is also prepared from by the use of microorganism. Gibberellic acid and gibberellins are also prepared by the use of microorganism. Lactic acid is also prepared from by the use of microorganism, and lactic acid it is used as a preservative in foods, in leather production and textile industry. Various other forms of lactic acid are also used for other purposes. Next. Amino acid. Microbial production of lysine and glutamic acid has recently increased. Lysine is produced by microbial fermentation. In microbial production of amino acid, only the desired products are produced. And next. Alcoholic beverages. Alcoholic fermentation has been well known since ancient times, though little was known of the nature of the process. According to Louis Pasteur, yeast were the organism responsible for fermentation of the sugar into alcohol. The beer is also prepared by the use of microorganism. It is a fermentation product. Next. Wine is also prepared by the use of microorganism. Saccharomyces is used in the production of wine. Distilled spirit is also prepared by the use of microorganism. Brandy, rum, whiskey, natural spirit, all these are the products which are produced by the use of microorganism in the industry. Biopesticides. Several microbes, that is viruses, bacteria, fungi, are being developed as a suitable biopesticide for management of insects and nematodal pests. Some fungi have good potential of their use as bionematicides to control nematode pests of vegetable, pest diseases of vegetable, fruits and cereal crops. Some bacterial and fungal products are also used to control diseases of roots and shoots of the plant. These are some microbial biopesticides: NPV, NPV, CPV. These are different types of microbes used as biopesticide.